Good day, everyone. I am Dr. Udemba Ibukunolu, a research scientist with the Institute of Agricultural Research and Training, Obafemi Awolowo University, Ibadan, Nigeria. I'm here to give a presentation titled Dynamics of Total Carotenoid Content of Yellow Root Cassava Varieties During Garri Processing. Projected is the agenda of the presentation, running from the introduction through to the summary and recommendation. Cassava originated from South America. It has risen to become the second most important crop after maize in Africa, being a major staple for more than 40% of our population. In Nigeria, about 80% of the populace eat cassava and its products at least once a day. Some even consume it more than once due to its relative affordability amidst prevailing economic recession. Meanwhile, cassava, though rich in carbohydrates, is deficient in many micronutrients, including vitamin A. This has led to the increase in the rate of vitamin A deficiency among consumers and producers of garri of cassava, resulting to partial or total blindness, increased susceptibility to diseases, and high rate of demise of children and pregnant women. To combat this, using cost-effective measure, cassava has been biofortified, leading to the development and release of some yellow cassava varieties with enhanced total carotenoid content. Unfortunately, cassava is rarely eaten raw, but is usually processed into diverse forms prior to consumption. In Nigeria, 90% of the produced cassava is usually processed, while 70% of the processed cassava accounts for garri production. Notably, carotenoids are naturally intracellular, while multi-step garri processing procedure can increase their availability and degradation. The degradation is also enhanced by the instability of carotenoids due to the presence of double bond in their molecular structure. The beta carotenoid, which is also the major constituent of yellow cassava variety, is very sensitive to light, oxygen, and heat, making it vulnerable to loss during processing. All this will influence the total amount of total carotenoid content retained in biofortified garri after processing for end users. Also, there are little or no information on changes in total carotenoid content of some elite biofortified cassava after various garri production steps. Meanwhile, understanding of this trend can help to set breeding targets appropriately, improve processing technique to reduce the carotenoid loss, and also help in varietal selection for propagation and adoption. This study was therefore conducted to quantify total carotenoid content in the fresh storage, root, intermediate product, and garri during processing to address this research gap. To achieve this, 3 yellow cassava varieties that are released in Nigeria were planted at the experimental site of the Department of Crop and Horticultural Science, University of Ibadan, Nigeria. They are the first 3 that are projected here, and a white root variety, which is the 4th, was used as a check. These were planted using a spacing of 1 meter by 1 meter with a total population of 30 plants per plot. The experiment was led using randomized complete block design with 3 replicates. 12 months after planting, the first storage roots were harvested and processed into garri following the procedure described by James et al. 2012. The first storage roots were peeled, grated, fermented for 48 hours, bagged, arranged and pressed using a metallic hydraulic jack to ensure simultaneous dewatering and fermentation for a duration of 24 hours. Subsequently, the dewatered mash were pulverized and garrified. At the iCheck Laboratory Lab of International Institute of Tropical Agriculture Following the procedure described by bioanalysts, the study was conducted in 2 seasons. Data collected were subjected to descriptive statistics, analysis of variance, and correlation analysis using the SAS software. Meanwhile, significant means were separated using least significant difference of means at 5% level of probability. Now to the results and discussion. Presented in Table 1 is the result showing the combined analysis of variance for the effect of variety, season, and processing stages on total carotenoid content of 4 cassava varieties during processing into garri. It was observed that all the factors and their interaction significantly influence changes in total carotenoid content during garri production. The mean total carotenoid content of the 4 cassava varieties at different garri processing stages are presented in Table 2. The effect of each stage of garri production on the TTC of the garri varies significantly, with the highest total carotenoid content recorded after garrification, while the least was obtained after grating. Total carotenoid content of the 4 cassava varieties at different stages of garri production is summarized in Figure 1. Notably, the white cassava variety had no total carotenoid content, which corroborates the earlier submission of Gomez et al. 2016 that most white varieties of cassava are devoid of carotenoids. Meanwhile, the presence of total carotenoid content in the yellow cassava variety suggests that they are veritable tools for combating vitamin A deficiency when consumed. The variety 0593 has the highest total carotenoid content after each stage of the processing, which was only significantly at par with 1368 after fermentation and dewatering. After grating, the carotenoid content of all the cassava variety of all the biotified cassava variety decreased. Now, carotenoids are naturally intracellular. Disruption of the tissues of the cassava through maceration and grating expose the inherent carotenoids and leading to the oxidation by the oxygen in the grater and the environment. This makes them vulnerable to be lost through leaching in the water from the cassava during grating. After fermentation and dewatering, all the total carotenoid content in the 3 yellow cassava varieties increased. This could be as a result of the production of additional carotenoids by the lactic acid bacteria and yeast which are present during fermentation. Also, softening of the cassava tissues during fermentation might have also increased the rate of release and availability of some carotenoids that were locked up in the cassava tissues. Dewatering also reduced the moisture content of cassava to about 40 to 50%. Therefore, this will also have led to the concentration of the carotenoids in the dewatered mash relative to the same quantity of the fresh root or grated mash. Garrification had different effects on the carotenoid content of the 3 yellow cassava varieties. Garrification led to increase in the rate of carotenoid content of 0593, suggesting that this variety has high carotenoid retention capacity after exposure to heat during garrification. Meanwhile, 0539 and 1368 have low carotenoid retention ability when exposed to heat during garrification. Also, garrification reduces moisture content by dehydrating them during the processing to as low as 7%. Therefore, the quantity of carotenoids in this sample after garrification would have also been concentrated and increased relative to the carotenoids in the fresh storage root, grated mash, or fermented and dewatered mash. It was observed when the percentage changes in the mean total carotenoid content of yellow cassava varieties during the processing was estimated. It was observed that 1368 had the highest deviation, the highest reduction from peeling to grating and from fermentation and dewatering to toasting compared to the other varieties. Also, it had the highest increase in carotenoid from grating to fermentation and dewatering. This suggests that the carotenoids that are present in these particular varieties are unstable, and their availability can easily be enhanced by cell destructive activities like the grating and fermentation and other processing activity during garri processing. Meanwhile, 0593 had the least deviation among all the evaluated varieties, suggesting that the carotenoids in this sample are very stable. The result presented in table 4 shows the Pearce correlation coefficient between total carotenoid content of the fresh storage route and product along the garri processing chain. Now, it was observed that there was significantly positive and strong correlation between TCC in the fresh storage route and the TCC content in the intermediate product and garri, suggesting that the amount of carotenoid content in the fresh storage route will influence the amount of carotenoid that is retained in the intermediate product as well as the final product, which is garri for end user. Observation and findings from this study, I would like to summarize that the total carotenoid content in the yellow cassava varieties fluctuated during garri processing. The biofortified cassava variety responded differently to each garri processing stage. Now, it was observed that the order of change in the carotenoid content was from garrification being greater than fermentation and dewatering, which was also higher than peeling, while grating had the least. Variety 0593 was most tolerant to loss in total carotenoid content at all the garri production stages. Also, this particular variety with the highest fresh storage route total carotenoid content will enhance the total carotenoid content of the garri and will ensure delivery of higher procarotenoids to end users, therefore standing as a great way of eradicating and correcting vitamin A deficiency among consumers of cassava. Propagation and processing of this variety is therefore highly encouraged and recommended. Meanwhile, the equipment used for grating yellow cassava variety should be modified to reduce loss at this stage. Thank you all for listening.